So this talk is all to do with an algae, or algae, you may call them. And this particular study is about the phylogeny of a particular gene from a particular alga. This is all of us. So the group consists of RMIT University, who did the majority of the microbiology, including growing the algae. The algae is presented there on the bottom right. That led to a bunch of phylogeny, but not before doing almost a full genome for this organism, just to find a set of, turned out to be 4 genes. The genes code for a biofuel producing gene called botriocoxine synthase. Now this fuel, botriocoxine, is an isomer of a ubiquitous cell constituent called squalene. We'll go into that. So we aimed to examine the evolution of squalene synthase enzyme to become botriocoxine synthase enzyme. Both of these hydrocarbons are triterpenoids with a carbon length of 30. And at least the botriocoxine is used by algae to float. By doing this during the Jurassic, it outcompeted its rivals in stagnant water by floating up near where the gases are. So we were aimed to obtain the genes, botriocoxine and squalene synthase, by high coverage sequencing around 100x, exhaustive blasting, iterative, and then selective assembly using SOAP on all of those hits to investigate a bunch of things: the gene number, structure, protein structure, and implied function of all those homologs. Squalene synthase and botriocoxine synthase are related, and I'd like to show you how. We wanted to know what order the genes developed in, and we found that we needed to develop a novel phylogenetic technique to actually even answer that question. I'm going to be discussing a thing called dimers, protein dimers. Homodimers is another word, and it's something that this study raised inadvertently, but very interesting. Okay, here's the structure of squalene. It's a C30 unit. So we've got 10, 10, and 10 equals 30. The 10 units of 10 are called terpenes, hence triterpenoid. Its functions are numerous, and it's usually excreted, being an oil. So it's in your sweat, shark's liver blubber. Cholesterols and ergosterols are not excreted, but they're intramembrane. The triterpenoid hydrocarbon botriocoxine is shown here, and its function, as I've mentioned, is to enable the alga to float. Now it can also be used to store energy, but so it's also used in shark's liver, interestingly, to help the shark liver, to help the shark stay afloat or buoyant. Now, botriocoxine and squalene are isomers, so a group has even managed to get yeast squalene synthase to make botriocoxine at low efficiency. It shows you that an enzyme derived from squalene synthase is quite capable of producing botriocoxine, so off we went on our hunt to find that enzyme. Here's the relationship between two products, botriocoxine and squalene, shown by virtue of the intermediate being common. The name of the intermediate is going to come up a fair bit in the talk. It's called presqualene pyrophosphate. It's quite descriptive, the name, and we shorten it to PSPP. Botriococcus braunii have had hundreds of millions of years to tinker with the evolution of these genes. So we expect that gene duplication was facilitated by this, and we want to know what the fewest number of changes would be to produce a new triterpenoid without affecting the production of squalene, because every cell needs squalene. So what we did is we estimated the genome size based on a paper that had appeared to be 160 megabytes, and we allowed for even more than that. Then we got some Illumina reads from memory. They were 100 bases long at around 100x coverage, and we did some paired ends, the minimum that we would need to get across introns, being low complexity regions. So we used local T-blast N to use ordinary squalene synthase from a green alga as the bait. And then we did some soap assembly, then GenBank BlastX of all the contigs that we obtained to figure out which were squalene synthase homologs, which were not, and then iterate, align with tools like bioedit. So single reads did the majority of this work, but I've explained what the paired ends were for. And so the exon-intron junctions, I got lucky that a postdoc I did was all about identifying splice junctions, so there was no problem there. And we obtained full-length proteins for not only this alga, but a lot of the model green algae like Chlamydomonas and Volvox. So we identified 4 genes. I called them Ayame triterpenoid biosynthesis 1 to 4. Ayame was the strain of algae that we used from race B, Botryococcus brownii. Okay, so another lab actually published the functions just as we put out our patent, and that was fine because we're not that competitive. And so they beat us to the right to produce these fuels, and our interest is also evolutionary. So we just included their 4 genes in our set, and so these analyses 7 to 12 all included their genes. So we aligned everything. We scored and distanced using actually using a technique that Mike will now cover.
Thanks, Bob. So whenever you want to compare sequences of amino acids, you're left with the question of what distance between the sequences means. We perform pairwise alignments using Needleman-Wunsch, weighing the distances using the PAM250 matrix. This is a stochastic technique, which models amino acid substitutions as a Markov chain and weights them according to how likely they are over a fixed span of evolutionary time. We ran non-metric multidimensional scaling, both to visualize the distances in 2D space, as well as to pre-process them for a subsequent run through UPGMA. We used n equals 5 for that analysis. These are both different ways to identify relationships between organisms. So you can see here why multidimensional scaling is so powerful as a visualization tool. MDS tries to preserve distances in the higher dimensional space as closely as possible in a lower dimensional space. In this example, 2D space. So the axes themselves don't have precise meanings, but what is important is that similar organisms should appear close to one another on these 2 axes. And indeed, that's what we see. We have some nice clusters corresponding to the A, B, C, and D groups, as well as model and other organisms. And it's nicely concordant with the phylogenetic tree we'll show you on the next page. You can also use the technique as a pre-processing or even as a compression tool. We also ran a non-metric multidimensional scaling analysis in 5 dimensions. Obviously, we can't directly visualize that, and generated an evolutionary significant phylogenetic tree, shown on the next slide.
Okay, I'm back now. Thanks, Mike. So the outcome was this phylogenetic tree, and whilst Mike might be able to describe better exactly what the distances mean, these distances are the key outcome of the tree. So those distances are meaningful. There's a scale at the bottom, and then the branching order was also rigorously produced. So this is not your kind of bootstrapped tree. This is the tree. This is the correct tree. So other kinds of phylogeny technique didn't actually work for this gene set, and it's only from long experience that I make that claim. In a paper we could do the comparison. So the B. brownii race B genes are in green. We've given them labels A, C, B, and D as in the multidimensional scaling, because the naming system used by the other group wasn't the same as ours, and so I just gave them random letters for each pair. Right, so you see that D evolved first, so that is actually the native squalene synthase of the organism. It then produced gene B, which is another squalene synthase. That gave it the freedom for homolog B to evolve, and so I'll go into what happened with A and C now. So here you have D producing B in an initial duplication, and then, as is often the case with gene duplications, instead of another one of those duplicating again, both of them duplicate at the same time, most likely. This can be something like a chromosomal duplication, or a duplication from one chromosome to another. So here we found that when they duplicated for the second time, they lost the anchor. So I'll go into these functions a little bit. When the squalene synthase became duplicated, it was able to reduce its production of pre-squalene pyrophosphate. Both of these functions are in the one organism, but it seems like the outcome of this whole evolution was to separate those two functions, because we find that when deduplicated, it created an enzyme, according to Nyhaus et al., Weiss, that only produces pre-squalene pyrophosphate. And this homologue over here, C, only produces butyrocoxene synthase. This is very interesting because it can mean that homologue A facilitated the evolution of homologue C. So we also did an analysis of the C termini, and we found that there was very well conserved intramembrane alpha helices. Now some would argue these can't be alpha helices because of things like cysteine and here amino acid K, but in general most of them have kept that and haven't actually mutated to the point where the intramembrane alpha helices are unfunctional, so it looks like they do have a function. There's also some membrane anchoring, affinity areas. So here's the crux of the talk. It seems that by developing special enzyme for pre-squalene pyrophosphate synthase and a separate enzyme for butyrocoxene synthase, that requires this insoluble product, pre-squalene pyrophosphate, totally insoluble in water, to diffuse through the aqueous cytosol over to butyrocoxene synthase, which is obviously not possible. So I've shown you these membrane tethers from the previous slide.
Mike here again. Picking up where Bob left off, PSPP is not free to diffuse through the cytosol as it is insoluble, but this is useful to its role as a membrane tether. To accomplish the transfer, these homologous enzyme structure-function reactions all involve a common active site structure, with the transfer inside squalene synthase being the most specialized. PSPP and squalene need to be made successively in the one active site, because PSPP isn't free to diffuse out of the enzyme complex. The active site has a hydrophobic pocket, and the transfer happens there. That means that it's not outside the realm of possibility that squalene synthase monomers actually may associate with each other, in the case of Botryococcus browni triterpenoid synthase, homologues A and C. Here you can see for comparison the homologous enzyme structures that involve proven homodimerization. And now back to Bob.
Here's just more evidence that they really do form excellent dimers. This one's the FPP synthase. You can go into that by slowing down the talk and having a good look.
We also did some functional studies, though without mutating things in vitro, by instead looking at the literature studies on mutagenesis of squalene synthases. We compared the primary structures of homologues A to D, and we noted the conserved residues that are specific to particular homologues, specifically residues that lie in active sites for either the first or second half reactions. These studies revealed synapomorphies that have been fixed in order to accomplish the splitting of functions. The specifics will be covered more in the accompanying paper. One example was the 5 mutations shown here, which lead to loss of squalene synthase function in homologue A. So where to next? Well, the full Botryococcus browni genome has been published by other researchers, and we would like to align all of our reads with the since published genome by the Chappell group. This future analysis will reveal new coding regions that their annotation missed, and is well preceded by interspecies yeast genome analysis. We've also started work on providing the community with BACs, bacterial artificial chromosomes. These are very large plasmids of the related Botryococcus browni races A and L. Sequencing race A next would be logical, as that race is likely the predecessor of race B by some 100 million years, and was responsible for pioneering the accumulation of oil in its intercellular space, albeit not squalene-related oil.
So we'd like to thank Pierre Metzger for supplying the strains of B. braunii. He supplied a bundle of them. We chose the best one that produced the most oil. Here's the back libraries I was mentioning. And the whole study was paid for by General Equity Building Society in New Zealand. Great blokes, and we'd like to thank them. Thank you.